So far, you learned that light reaction results in the formation of ATP, NADPH, and oxygen molecules. Now let's see how plants utilize these molecules to prepare food through the Calvin cycle. Oxygen released during photosynthesis is released into the atmosphere through stomata present on the leaves. While the ATP and NADPH molecules are used in the synthesis of carbohydrate from carbon dioxide and water. This phase is called the biosynthetic phase, as it involves the production of food. It is also called dark reaction. Dark reaction takes place in the stroma of chloroplast and is not dependent on light. Dark reaction, as its name suggests, does not necessarily take place in the dark, but takes place both in light and dark. Until the products of light reaction, ATP and NADPH, are exhausted. The American scientist Melvin Calvin used radioactive carbon-14 in algal photosynthesis and discovered that the first stable product formed in the biosynthetic pathway is a 3-carbon compound, 3-phosphoglycerate. However, only some plants showed 3-phosphoglycerate as the first product of carbon fixation, while others showed the formation of a 4-carbon organic acid, oxaloacetic acid. Plants that showed 3-phosphoglycerate as the first product of carbon fixation were called C3 plants, and the pathway was called the C3 pathway. Plants that showed oxaloacetic acid as the first product of carbon fixation were called C4 plants, and the process of carbon fixation was called the C4 pathway. Melvin Calvin, James Bassam, and Andrew Benson worked out all the biochemical reactions of the C3 cycle. The C3 pathway was named after Melvin Calvin as the Calvin cycle. The Calvin cycle has 3 stages: carboxylation, reduction, and regeneration. In carboxylation, a 5-carbon molecule, ribulose 1,5-bisphosphate, accepts a carbon dioxide molecule to form an intermediate 6-carbon molecule, which then splits to form 2 molecules of 3-phosphoglycerate. This reaction is catalyzed by RUBP carboxylase. Ribulose bisphosphate carboxylase also exhibits oxygenation activity. Hence, it is called ribulose bisphosphate carboxylase oxygenase, or simply Rubisco. This phase is called carboxylation because a carbon dioxide molecule is added. In the reduction stage, a series of reactions leads to the formation of triose phosphates, which act as precursors to glucose and other sugars. At first, 2 molecules of 3-phosphoglycerate is phosphorylated to 2 molecules of 1,3-bisphosphoglycerate by utilizing 2 molecules of ATP in the presence of the enzyme phosphoglycerate kinase. Second, the enzyme glyceraldehyde 3-phosphate dehydrogenase reduces 2 molecules of 1,3-bisphosphoglycerate to 2 molecules of glyceraldehyde 3-phosphate. 2 molecules of NADPH are used in this reaction. Thus, to fix 1 molecule of carbon dioxide to 2 molecules of glyceraldehyde 3-phosphate, 2 ATP and 2 NADPH are utilized. 6 carbon dioxide molecules are necessary for the formation of 1 glucose molecule. Hence, let's calculate for 6 carbon dioxide molecules. 1 carbon dioxide molecule results in 2 molecules of glyceraldehyde 3-phosphate molecules. Hence, 6 molecules would yield 12 molecules of glyceraldehyde 3-phosphate. Of these 12 molecules, 2 of them are used to make 1 molecule of glucose. The remaining 10 glyceraldehyde 3-phosphate molecules make up a total of 30 carbon atoms. These are involved in the regeneration of 6 molecules of ribulose bisphosphate, a 5-carbon compound, thus balancing the reaction. In the regeneration phase, 1 ATP molecule is utilized to form 1 RuBP. Hence, during the Calvin cycle, 1 carbon dioxide molecule needs 3 ATP molecules and 2 NADPH molecules for fixation. Therefore, the net utilization of energy for the formation of 1 glucose molecule would be 18 ATP and 12 NADPH molecules.